All right. Well, good afternoon, everyone. For those who are joining from the waiting room, welcome, welcome. Today we have a briefing on gene therapy for genetic hearing loss, which is hosted by the American Society of Gene and Cell Therapy. We're very glad to have you here. My name is Christina Mayer. I'm the Director of Government Affairs at ASGCT, and we are so grateful to have Representative Kevin Mullen, who is co-chair of the Bipartisan Bicameral Personalized Medicine Caucus, as our co-sponsor for this briefing. So today we have an esteemed lineup of speakers from the scientific and patient advocacy worlds. Before we get into that briefing, a few housekeeping items for our attendees. First of all, this briefing will be recorded and it'll be posted to ASGCT's YouTube channel within the next couple of days. So if you have to duck out a few minutes early, we know this is a very busy day on the Hill for the health folks. Or if you have a colleague who wasn't able to attend live, we'll be sure to send that YouTube link around so folks can catch up with the whole thing. Second, there is a Q&A button at the bottom of the screen where we invite you to submit any questions you have for our speakers. That can be done anonymously if you prefer. And we'll collect those questions and we'll have a roughly 15-minute panel discussion at the end of the briefing. So as I said, we are so pleased to have Congressman Kevin Mullen as our co-sponsor today. He recorded a brief video welcome, so I will hand it off to him.
Thank you all for joining today's briefing on gene therapy advancements for inherited hearing loss. I'm Congressman Kevin Mullen, and I represent California's 15th Congressional District. It's an honor to co-host this event with the American Society of Gene and Cell Therapy. I'm glad you can all join us to learn about the groundbreaking medical advancements being made in the hearing health space. As someone who lives with genetic hearing loss, I need hearing aids to function on a day-to-day basis, and I know firsthand how expensive these devices are and how difficult it can be to have hearing care covered by insurance. And I'm not alone. In the United States, about 2 to 3 out of every 1,000 children are born with some level of hearing loss. This means roughly 20 to 30 babies born each day grow up facing the potential negative effects of hearing loss. It puts individuals at higher risk for both mental and physical health problems due to social isolation and higher rates of loneliness. Adults with hearing loss are at a higher risk of dementia, accelerated cognitive decline, depression, anxiety, and falls. That is why this work to advance cell and gene therapies for hearing loss is so important. Researchers are discovering pathways that could change the lives of countless individuals, turning what felt like science fiction into a reality. But there are no FDA-approved therapies yet, so we can't take our foot off the gas. It starts with fully funding the NIH and increasing funding for institutes like the National Institute on Deafness and Other Communication Disorders. NIH-backed research helped lead to the discovery of a gene that, when mutated, is responsible for up to 8% of congenital hearing loss. Clinical trials are currently underway to treat hearing loss caused by that gene's mutation. Fully funding the NIH ensures researchers continue discovering new causes of hearing loss that future therapies can treat. And it will ensure that the U.S. continues to lead the world in medical advancements. I'm proud to be a champion in Congress for the hearing loss community. During the fiscal year 2027 appropriations process the House recently completed, I led requests to increase funding both for the NIDCD and the hearing restoration research conducted through the Department of Defense. And I'm the sponsor of 2 bills to make hearing aids more accessible and affordable. The first is the HEAR Act, which would require traditional Medicare to cover hearing aids. The second is the Hearing Aid Assistance Tax Credit Act, which would create a tax credit for those who purchase hearing aids. I'm also the co-chair of the Personalized Medicine Caucus, which is focused on educating congressional offices about the value of investing in treatments tailored specifically to each patient. Thank you again to ASGCT for putting on this event, and thank you to the panelists for taking the time to share their expertise about this exciting space. Thank you.
Right, so thank you again to Congressman Mullen for his work in that space and for that really hopeful message about the power of biomedical research. So moving on to our panelists, I'd like to first introduce Dr. Matt Porteous, who is ASGCT's president-elect. Dr. Porteous will introduce himself further and kick things off, and then we'll hear from each of these speakers in their turn, and they'll introduce themselves further when it's their turn to present. So with that, Dr. Porteous, I hand it
Thank
off
you.
to you.
Thank you. Yes, my name's Matthew Porteous. I'm a professor of pediatrics and the Satarja Professor of Definitive and Curative Medicine at Stanford. First, I am the president-elect of American Society of Gene and Cell Therapy and I'll take over the president of the society in May. Want to thank Representative Mullen for his passionate advocacy for both NIH funding, uh, cell and gene therapy funding, and specifically for research to improve the lives of people with hearing loss. So my task over the next few minutes is to introduce briefly the society and what we do, and then broadly the field of gene and cell therapy. Next slide. So the American Society of Gene and Cell Therapy is the largest society in the world whose mission is to advocate and support research and development in the gene and cell therapy space. We have over 6,500 members, and this year is our 30th anniversary. Our membership is broad and consists of scientists, physicians, patient advocates, and other professionals who work across universities, hospitals, governmental agencies, foundations, biotech, and pharma. And today's expert speakers who will follow me represent many of these communities. We advocate for improved patient education, global outreach, policy and advocacy, and education, in addition to supporting the scientific and clinical progress in gene and cell therapies. Next slide. So quickly, in our title, we have two names, gene therapy and cell therapy. And gene therapy is a process in which we give a genetic material, whether it's DNA or RNA, directly into the body. Actually, that's called in vivo gene therapy. And actually, for hearing loss, we will hear about the in vivo approaches. Or pieces of DNA are directly modified in the body. But complementary to gene therapy, where the drug is given to the person, there's cell therapies in which cells are modified outside of the patient or the person, and they're modified using different techniques and then given back to the person to treat their disease. These are There's multiple, and we'll get to this, there's multiple approved, FDA-approved therapies for both gene therapies and cell therapies. The most common of which cell therapy is chimeric antigen receptor T cell therapy to treat blood cancers. Next slide. One of the reasons that gene and cell therapy have such power and have the potential to be one-time cures for diseases is that they target the pathophysiology of the disease at its root cause. That is, for inherited or congenital forms of diseases such as hearing loss, it is often caused by a change in the DNA sequence of a cell. And gene and cell therapies can address that disease or characteristic causing change directly at its root cause. And by directly addressing it at its root cause, not only is all the downstream effects of the change in the DNA counteracted, but because the DNA is carried in the cell for the entirety of that cell's lifetime, this can have years, if not lifetime, durability. The positive impact of this is both direct on the patient's life, but also very positive, indirect, and intangible costs. For example, a person with a disease, because of their disease, may not be able to pursue their dreams in their life, whether that's professionally or in their hobbies. And now, by addressing their condition at the root cause, they're now able to pursue their dreams. Next slide. So, as I mentioned, there are various types of gene therapies. And because we have a rich toolbox, we can design different gene therapies for the specific conditions. And again, you'll hear from our experts about how different technologies are being applied to inherited hearing losses. And again, this can include in vivo gene therapies in which the genes are introduced directly into the patient, or in ex vivo gene therapies in which the cells are removed and then given back. Gene editing and CRISPR gene editing, an incredibly powerful technology, it can be used either in vivo to directly change the sequence of the DNA in a cell, or can be used to modify the DNA sequence of a cell outside the body. And it has the ability to change the DNA of a cell exactly where you want. And the term implies this precision, like we can edit a document on our computer. Next slide. So the field is exploding, and we now have 41 gene and cell therapies that are approved in the United States. We have 27 RNA therapies and 70 non-genetically modified cell therapies, shown in the pie chart on the right. And next slide. And there are thousands more in the pipeline, ranging in with, if you look into the diseases for which clinical trials are being performed, you see these range from cancer to rare genetic diseases, neurologic diseases, sensory diseases, and the list goes on and on. These span the entire gamut of clinical trials, from early phase 1 trials to phase 2 and 3, and then even pre-registration trials. And in the next slide, we can see the listing of these approved cell and gene therapies. So cell therapies listed under ex vivo, gene therapies listed under in vivo. And you can see they all were approved with a 2 in the 21st century, and many now have been approved with the third number being a 2, so just in the last decade. And this explosion in approvals will continue to occur over the next several years. Next slide. So before, I wanted to thank you for attending this briefing. We look forward to your questions and answers. And before I turn it over to my expert panelists in this specific condition, we have a brief introductory video into the genetic causes of hearing loss. So thank you.
Let's talk about genetic causes of hearing loss. More than half of hearing loss cases present from birth have a genetic cause. This is known as genetic hearing loss. The human ear is complex, and there are many parts that work together to help us hear. There are hundreds of genes that help these parts of the ear develop and function. And when one gene doesn't contain the correct instructions, hearing loss can occur. Genetic hearing loss may be caused by changes in the genes, known as gene variants, which affect how the ears work. There's thought to be hundreds of types of genetic hearing loss affecting different genes and different cells in the ear. The only way to confirm the cause of hearing loss is through genetic testing. Genetic testing, along with current standard tests, may allow for more personalized care and help inform the choice of hearing aid, cochlear implant, use of sign language, and other forms of visual communication, or a gene therapy. Human beings develop the majority of their language skills by age 3, so it is important that people are informed about their options to maximize the primary years of language development, may it be listening and spoken, or any form of visual or sign language. Gene therapy is a new potential treatment for hearing loss. These therapies are generally developed to address the underlying genetic cause of a condition. In this case, it may help enable or preserve hearing capabilities. Because the genetic causes of hearing loss vary, each genetic variant is likely to require development of its own unique therapy, meaning a single gene therapy will not treat all types of genetic hearing loss. An example of a gene therapy approach being studied for hearing loss seeks to correct a non-working otoferlin gene, or OTOF gene for short. This gene produces otoferlin protein, which is key for sounds to be transmitted from the ear to the brain. Gene therapy uses a modified virus called a viral vector to deliver a working copy of the OTOF gene to the inner ear. There are several different surgical procedures that are being investigated with the common goal of delivering the gene therapy directly to the inner ear. Scientists know that viruses are good at getting into cells, so they have learned how to remove the harmful parts of the virus so only the working genes are delivered. Once in the cells of the inner ear, the working OTOF gene may help restore proper production of the otoferlin protein that was lacking within these cells, with the goal of enabling hearing capabilities. Children with a cochlear implant in both ears may not be eligible to receive a gene therapy. However, research will continue to evolve. The decision to explore potential treatment options for hearing loss is an individual or family decision. While scientists have worked for many years to develop gene-based therapies for several types of genetic hearing loss, it is important to understand that gene therapy must be researched in clinical trials to determine if it is safe and effective. For more information about gene therapy for genetic hearing loss, visit patienteducation.asgct.org.
Okay, I'd like to turn it over to Kristen Wolf, who's the Associate Vice President for Commercial at Eli Lilly Gene Therapy, to discuss more specifics around gene therapy for hearing loss.
Thank you so much, Matt. And a very sincere thank you to ASGCT for the invitation to be a part of this briefing this morning. Thank you, everyone, for joining, and for those who are listening online, as I always like to consume content after the fact. My name is Kristen Wolf. I lead a commercial for Lilly's genetic medicines unit, so the specific part of Lilly that is focused on genetic medicines. I have several slides today actually to pull through some of the themes that you just saw in that really well done video. Next slide. So I was asked to focus specifically on clinical development for gene therapies. For those who are maybe not as familiar with drug development, when we say clinical development, we mean development of an investigational therapy in humans. So there's a lot of work that goes into understanding, characterizing, learning more about a potential medicine before you ever even dose the first patient in a clinical trial. That's usually called discovery, or the research phase. And then you transition, when you get into the clinical trial, to the development phase. And then hopefully what we're all working toward, and specifically what I spend all of my time doing, is remembering that FDA approval, regulatory approval is critical, but that's not the finish line. For a lot of us, that's the starting line to then actually get a working, safe medicine to the people that want it. Next slide. These figures were touched on a little bit during Representative Mullen's introduction, as well as in the video, but it bears repeating. These are small populations. There's no doubt about that. But there is high unmet need here, and even higher than that unmet need is the potential transformative impact of a gene therapy, a therapy that really addresses the root cause of the condition in the first place. It's that potential to affect transformational change for an individual, for their family, for their broader community, that really drives the commitment that you see, not just from Lilly, from all the companies that are in this space, and really from all of the healthcare stakeholders that are in this space, physicians, patients, patient advocates, healthcare systems, sort of the institutions that surround the physician and really support them, policymakers, and like yourselves, and governments. So we really wanted to sort of highlight that commitment. Small populations, but high unmet need. You've heard the statistic: 1 in 500 infants is born in the US will go on to, or that is born with a hearing condition, or will develop it very early on in childhood. I think Representative Mullen said something like 30 babies a day. In 2024, that would have been over 7,000 babies that were born with a hearing condition. And 80% of those hearing conditions have a genetic cause. Most people, as we heard about in the video, genetic testing is necessary to confirm the diagnosis. Most of those 7,000 babies will never know not only that they have a genetic cause to their disease, but specifically what that genetic cause is. And that knowledge can really help unlock potential treatment options. So the theme that kind of is the undercurrent of my presentation is the importance of access to that genetic testing for people. There are interventions on the market that are incredibly helpful, and I've actually seen a lot of technological advance over the last not only decade but 2 decades. Hearing aids and cochlear implants are available to those who are interested in using them, those who have hearing loss. But I think it's as much progress as those technologies have made, it's important to point out these are not replacements for natural or physiologic hearing. And they are associated with long-term maintenance costs and long-term obligations that can continue to connect individuals who use them to the healthcare system. Next slide. There are several approaches. So you just heard the why. That's what that slide was. The why. Why are people so interested in doing this? Here's the how, and the how is really where rubber hits the road. So I've highlighted this is not the universe of gene therapy approaches for genetic hearing loss, but these are the ones that are being employed today, some to really great success and others that hold a lot of promise, but where we really haven't fundamentally unlocked the basic scientific questions. And it's not, again, just companies like Lilly that are trying to unlock those basic scientific questions, but as Matt was talking about, this is where institutions like the National Institutes of Health can really help with the basic science research that is the foundation of drug development. Gene transfer and genome editing, these are the two types of genetic therapies that in other disease states have actually gone on to lead to successful medicines that are available to patients today. Gene transfer is packaging a correct working copy of the faulty gene and delivering it to the cells. Editing is a similar concept, but instead of sending the full gene to the cells that need to utilize it, it's rewriting the existing DNA that's there. Think of it as opening up a Microsoft Word document and Control F to find an error and then being able to Control R to replace. Stem cells, just as a quick primer, stem cells are these naturally occurring blank slate cells that can then go on to become all the different cell types in our body. So stem cell approaches often try to use blank slate cells and direct them to become the specific type of cell that is damaged in a particular hearing loss condition. iPSCs, forgive the fact that I used an acronym, but it stands for induced pluripotent stem cells. This is actually starting sort of at a different point in the process, still stem cells, but taking ordinary cells, coaxing them into going back in their development to become these blank slate cells, and then directing them to become cells that are needed to replace the ones that are damaged. Exosomes describes a type of technology. So cells have sort of an innate signaling technology between each other to send each other messages. This is redirecting really, those messages that cells naturally use to communicate to signal directly to damaged cells, like the inner cells of the ear, to stimulate repair and regeneration. And then tissue engineering or cell scaffolds, this is really reconstructing the damaged structures of the inner ear from the ground up. Scaffold is sort of the perfect evocative word to use here as you imagine it. So two of these technologies are tried and true, so to speak, in other conditions and are now being applied in the hearing loss space. And the others we're still working through some of those challenges. And the next slide actually goes into some of those challenges. And I'll pick up the pace here and just say, so you saw in the video, you'll see here again, the inner ear is encased in bone. It is actually a very difficult set of structures to access. And so that is the primary challenge with which early drug development struggled. We've made a lot of progress accessing those structures, getting these technologies to the cells that can benefit from them, but we still face quite an additional list of challenges. Optimizing how well a gene therapy targets the right cells, how well, when it targets those cells, it can actually deliver things like the functional copy of the gene into that cell, and the gene can then use that DNA. Minimizing potential side effects, this is something I know everyone in this field really thinks about. And then recognizing that when it comes to addressing hearing loss conditions at their root, Not every genetic causes a single gene. You could have multiple genes that go on to cause a condition. And how do you address that kind of a genetic etiology is something that scientists are still working on. Last slide. I won't go through this in detail, but this is just to say 10 years ago, the slide would have largely been blank. There was not a lot in clinic, actually in clinical trials with humans in the hearing loss space. And now there's certainly been a concentration in gene therapies that are focusing on OTOF because it's a well understood gene, genetic mechanism, but you're starting to see branching off into other types of hearing loss causes with these gene therapies. And I almost guarantee that if we were to come to you again in a year or 2 years, we would need more than one slide to really describe what's going on here. So the pace of progress is picking up, and we really appreciate all of the efforts to help that pace accelerate faster. Thanks so much.
Turn it over now to Dr. Hastings, who's the Pfizer Upjohn Research Professor of Pharmacology at University of Michigan Medical School, who will discuss a different way of addressing genetic hearing loss.
Thank you, Matthew, and thank you for inviting me. It's my pleasure to have a few minutes to talk to you today about precision medicines for hearing loss. And more specifically, I'm going to talk about potential for small RNA molecules for treating hearing loss, so a little different modality than what you've heard about so far. Can you go to the next slide? Next slide. Subtitle. So nucleic acid therapeutics is a very broad class of therapeutics, and I've listed most of them here, not all of them, but the ones that are the major players. Gene therapy is the one you've heard about now, and I included viral mediated gene delivery here. And then the rest of these are largely RNA-based therapeutic approaches, and I have circled there the class of oligonucleotides. So I'm going to briefly tell you about this group. If you go to the next slide, I'm showing you here the FDA-approved commercial RNA-based RNA medicines from the previous slide. And I'm not including the viral-mediated gene delivery here, just the RNA-based medicines. As we've heard, there's currently no precision nucleic acid or RNA-based medicines for hearing loss. However, what I'd like to show this is that there is a remarkable success with this and a growing success of these types of nucleic acids therapeutics. And what we have found through our research is that there's a remarkable ability to prevent or recover hearing loss. And this has largely been done in animal models and, in addition, a growing number of clinical trials. And this gives hope that there will soon be medicines that effectively treat the condition under these different types of approaches. And I think today, giving you an example of this will really highlight the promise of precision RNA medicines. And so I've decided I would tell you an example from my own research of the development of a precision RNA medicine to treat a mouse that was genetically engineered to have a human form of profound congenital hearing loss, a genetic form of human hearing loss. And we made a nucleic acid molecule called an antisense oligonucleotide, or sometimes called an ASO, to treat these mice. But the same molecule could also be used in humans because the gene we put into the mice is identical. Can you and so the antisense oligonucleotides are sort of this grouping on the left. So by way of a brief background, antisense oligonucleotides, also called ASOs, are just very short pieces of nucleic acid that work by binding specifically to a piece of RNA. And RNA is the message-bearing output of our genes. The sequence that you make the oligo, and the oligo is shown there in green, directs it very precisely to a single specific location on the RNA. So they're very unique in that they act like a beacon and they go exactly where you want them to go. And if you know where to direct it on an RNA, you can modulate the way the RNA message encodes a protein. In the case of a disease or a pathological condition like hearing loss, modulating a defective gene product in the right way can correct its defect and restore proper protein expression. And one advantage of these types of short nucleic acids is their simplicity. in all the experiments I'll talk to you about today, and a lot of the drugs that are out there for different conditions, these are just naked modified nucleic acids. They're short, they have manufacturing advantages and such to other types of modalities. On the next slide, by way of background, the disease that we tackled in my lab was, or the syndrome, I should say, was Usher syndrome type 1C. It's the leading, Usher syndrome in general is the leading genetic cause of combined hearing loss and blindness, and type 1C is a variant, a Usher syndrome causing variant in a gene called USH1C, type 1C. Children never develop the ability to hear, and then they have vestibular and balance problems. And later in life, they go blind, and typically in their teenage years. In the next slide, I have just outlined in a cartoon here, when you introduce this genetic variant into the mouse gene, the USH1C mouse gene, they have very similar problems as in humans. They never develop an ability to hear, and they have very severe balance problems. In this cartoon, those oligos are just in green on the top, and we weren't sure how to really treat the mice, but we figured we should treat them early, and we just gave them a shot, an intraperitoneal, like a sub-Q injection into their abdomen. And what we found, if you show the video on the next slide, on the bottom, you can see a mouse that has this variant, this USH1C causing variant introduced into them, and they run in circles because the vestibular organ next to the hearing organ in the inner ear is also defective, and so this causes a balance problem, and they run most of their time are running in circles when they're awake. And that gave us a really good rubric. If you go to the next slide, when we treated these mice on the lower right now, you see an Usher mouse that's been treated with our antisense oligonucleotide, and you can see that it's really indistinguishable from, on the top row is the mice that don't have the variant causing Usher syndrome. So we had this remarkable recovery of vestibular imbalance, that the mice actually never developed any vestibular problems, and so we consider them cured from this single injection when they're first born. On the next slide, we went on to test hearing of these mice using the same type of hearing test that humans get, a measure of auditory and brainstem response to sound. And these studies are just basically, they were carried out by Jennifer Lenslab at LSU, and the waves indicate ability to hear. On the next slide, here's the auditory brainstem response. On the left is a mouse without the genetic variant. In the middle is our diseased USH1C mice. You can see there's no response, and on the right is the mice that were treated with our oligo, indicating that they're now having this response. So that was really exciting. On the next slide, we worked with Gwen Jeliak at Harvard, who is working on a gene therapy like you heard about for the AUTOF on the left. And so we worked with her to compare how the short antisense oligos looked comparatively to a gene therapy. And this is a busy, fairly busy graph, but basically what it's telling you is that they really are pretty comparative in their effectiveness and their longevity. They work for quite a long time after a single injection directly into the inner ear. On the next slide, I just put this in here to talk to you about how delivery to the inner ear has some really interesting advantages in terms of treatments for these modalities. There's many different ways that seem to be effective, even just on top of the tympanic membrane. But also we did direct delivery into the amniotic fluid of pregnant dams and found that also prevented vestibular dysfunction. And so there's really some interesting benefits, especially in hearing loss where you might need to, congenital hearing loss where you might need to treat early. On the next slide is just a summary of the work that we did and learning a lot really about these types of oligos and their ability to treat hearing loss, vestibular function. On the next slide, I just wanted to say that for these types of modalities and treatments, what we really need is targets. And what we have here are a lot of targets. There's over 156 hundreds, as we heard earlier, of genes that we could target with clever designs. And as I've shown you, there's a lot of high potential. On the next final slide, again, this is such an exciting area, unprecedented potential. And what is the holdup? First of all, we need to understand the target. So we need more basic research. And this funding comes from the NIH as well as development. I've highlighted here that, as the senator said in his opening remarks, NIDCD funds most of the hearing and communication disorders like hearing loss. And you can see it, you can't even see it, such a small sliver in the percentage of funding from compared to other institutes at the NIH. And then it's hard to see, but this table just indicates that, we're flatlined or going down in our NIH funding. And so while we have some really amazing abilities to really make a difference in treating these, getting them through to the finish line, including for this ASO for Usher and the gene therapy is really just a matter of commitment and getting it through. So that's all for me. Thank you for your time.
And we'll conclude our expert panelists with Janet DeSorge, is the Executive Director of Hands and Voices and a strong patient advocate for the community of people with hearing loss.
Hi, everyone. I'd like to thank the organizers for this opportunity today, as well as Representative Mullen. For those of you here who are attending, thank you so much for your time. Hands and Voices is a parent-led organization specifically for families on the journey of raising a child who's deaf or hard of hearing. Our motto is, what works for your child is what makes the choice right. We know that the paths to success in the journey of deafness is very unique for every individual child. I'm sure you can relate to this idea that there are moments in your life that you will never forget. Sometimes good things like the birth of a child, graduation, wedding day, other things that can be more challenging. I will never forget the moment that an audiologist walked through the door after testing as I was holding my daughter, Sarah, in my arms and delivered the news to our family that Sarah was deaf. The first questions and fears that come up in the minds of families during this time can run the gamut from, will she be invited to the prom when she's in high school? How is she going to learn to communicate? What language will she use? Will she be bullied in school? How will she have a sense about who she is as a deaf individual? Of course, there's also medical questions that come into the minds of families of, why did this happen? What is the cause? For families who have children who are deaf or hard of hearing, over 90 to 95% of those families come from are hearing themselves. We have no particular background in understanding how or why this happened. But I do want to tell you that at this very moment, it's not just a medical moment for us. It's a broad-based feeling of thinking about human beings. In essence, the very foundation of what makes us human is language and communication. Families are faced with many decisions along this path, often in a very time-sensitive manner. It was shared earlier that the period for language acquisition is through the first 3 years of life. So while today in our system of care, 99% of babies are now screened in the hospital for hearing difference or hearing loss, the path, so we're finding out earlier, which is better, but the decisions that families need to make are still really critical, and family needs space and time to make informed decisions about the things that they'll need to think about for their deaf child. That's what I'm going to just focus on primarily here in the few minutes that I have. When it comes to gene therapies as they're emerging, I can tell you as a hearing parent, one of my first questions is, can you fix this? in the history of deafness, until the last couple of decades, that question was always answered with no, but there's other things that we can do to help your child be a full, well-adjusted, successful human being in this world. I think the conversation about gene therapy in terms of navigating the current complexity of this type of decision is, in general, this isn't really a for or against conversation. Gene therapy is not a simple yes/no decision. We know right now even the percentage of families that are even eligible for the clinical trials are very few. It's a very small percentage right now with otoferlin. However, I'll tell you, every family who has a child who's deaf or hard of hearing may be getting the phone call from their mother-in-law who just saw the news of the clinical trials being really effective in terms of restoring natural health. So this is of vital importance for all of us parents and patient advocacy organizations to be navigating the complexity of this. When we think about informed decision making, you can break it down into things like accurate, balanced information that's timely, access to diverse perspectives in this, whether it's medical professionals, deaf community, deaf culture, deaf and hard of hearing adults, and other parents. The time and the support that families need in order to process these kinds of decisions are a part of informed decision making, and then a system of care through parent organizations, other parents, deaf, hard of hearing, culture, and community is the idea of accepting the idea that families will make different choices that are right for their child. When I think about the influence of those in this room today in terms of policy thinking directly, we've been talking about what NIH funding enables, scientific breakthroughs, understanding the variability in outcomes, which is where we're at right now in clinical trials, the long-term safety and effectiveness. These are some of the things that families really need to understand and know, and that this funding can help support. NIH funding also supports beyond this space language and communication outcomes and family-centered practices. When we think about what investing in these areas and in this space, it's both innovation and informed choice. And this brings us to the full circle, which is for families, we want to know what options exist. We want to know if information is trustworthy, and support will be consistent no matter what the decisions we make. Finally, our role is not to tell what families to do, but to make sure that they truly have options in which to choose from. So I'm so grateful for this conversation today, for those of you who have areas of influence when you talk about this. And we want, for families who have children who are deaf or hard of hearing, we want what every parent wants, which is for our children to thrive, to communicate, to have an effective education, no matter where our families come from rural, urban, whatever socioeconomic or ethnic background, to make sure that the system works for all kids. So I thank you for your time today, and thank you to all the other presenters who have shared as well.
Thank you, Janet. We really appreciate that. And thank you to all of our presenters today. We will get folks pinned into kind of a panel view for this Q&A period. So starting off for you, Janet, so one of the things you mentioned was that families have to dig through a lot of information, and obviously we live in a world where there is a lot floating around for folks to find on social media, on government websites, kind of all and everything in between. And for example, ASGCT has a patient education program where we have dozens of videos to try and help give patients some of that trustworthy information, including the video we played earlier. But how do you help parents sort of decide what's trustworthy and find those trustworthy resources?
That's a good question. I think this is a huge conversation in any space, whether it's audiologists talking about the support for families, early intervention, education. You can try to safeguard information, for instance, like how much information should you give families at the time of diagnosis? I'm telling you, families will go home and hit the internet. So we know that information, we're living in an information. So how do we know it's trustworthy? I think it goes back, of course, to the source. So families need to be able to understand the source. When it comes to deafness, there is a lot of factual information and research evidence-based practice, but there's also very unique outcomes for each individual child based on research. And so it's both an objective and subjective. So the way we talk about it is to make sure you trust the source you're getting information from. And also, if you have a question, ask a variety of people that same question so that you can get different opinions and feedback in that way.
If I could add to Janet's point about trust, is I would highly recommend that the ASGCT website is one of the trustworthy sources of information. That is our mission, is to be trustworthy. And I would also say that of course, people aren't going to stop there, but I think it should be one of the stops on people's journeys to understand where really are we in the cell and gene therapy space for a specific condition. So I hope, if nothing else, taking away that we can be one of the trusted stops in that internet journey.
I would just like to add that I think ASGCT did a wonderful job in getting feedback and perspective from a lot of patient advocacy groups, deaf, hard of hearing individuals, and that's obviously reflected in the video, which is great. I'll put in a plug as well for handsandvoices.org. Yep. We have a whole genetic education information section, and we know that the path to information and decisions about therapy or cell options comes from genetic testing itself. And so we've spent a lot of time in terms of thinking about families making informed decisions about genetic testing itself too. So yeah, thank you, Dr. Porteous. I agree with you.
Thanks for that. So Kristen, I'll kind of pitch this to you, but if anybody else wants to chime in as well. You mentioned that slide about what's in clinical development now would have been blank a few years ago, and in a few years ahead, it's going to be, multiple. But from a kind of commercialization standpoint, what are the sort of biggest real world challenges you and other companies are facing as you try and take these, promising lab discovery, things coming out of Dr. Hastings' lab and all these other places into those clinical stage opportunities for patients?
Absolutely. Thank you for the question, Christina. And I invite all the other panelists to chime in as well. I would start by saying, and this is a callback to some of the remarks I made during the presentation, one of the biggest challenges that we face to date is right now there are no there are no sort of medicines on the market for patients who have hearing loss, let alone genetic hearing loss. And so there's not a lot of genetic testing that goes on because there isn't a difference in the way that you would necessarily, or in the treatment options to which they would have access. So those two things go hand in hand, low genetic testing with sort of a lack of available therapies. We hope that the reverse is true, and we are working with stakeholders at the state level, at the federal level, across patient advocacy organizations, as well as healthcare institutions to help facilitate the opposite being true, which is when there are genetic medicines that are FDA approved and on the market, that genetic testing rates go up. But we know that it will take work. And I think that is a challenge that is often undersold, but has a dramatic bearing on if people are even aware of the fact that there are clinical trials right now and hopefully one day FDA approved therapies that may be appropriate for them to consider. And that's all we want, is an environment where there's enough information that's presented to the right audiences at the right time that they can use, as Janet said, to make the best decision for their family. And everyone's decisions may be different, and that's an ecosystem that we really strongly support. I think the other thing that I'm conscious of, and please take this as from Kristen rather than from Lily, is that we are talking about a disease state, being deaf or hard of hearing, that doesn't necessarily involve a lot of costs to the healthcare system right now. And I mention that because that can affect how insurers, Medicare, Medicaid, commercial insurers, think about how valuable the therapy is. And that is we think that the old ways of looking at value, how much should you value a therapy, really aren't fit for purpose when we're thinking about a gene therapy, let alone a gene therapy for a condition like hearing loss. So that's a lot of work that we're focused on now, in the years before a gene therapy really comes to market, to make sure that a broad range of value considerations is taken into account. The value that patients themselves would accrue to having access to these medicines, that their families would value in the context of educational attainment. That sort of broader perspective of value is meaningful across all disease contexts, but especially for gene therapies and especially in the hearing loss space. So that's what I would highlight.
Appreciate that. Question from the chat, which is kind of talking about how gene therapies might play with other interventions that we have today, hearing aids, cochlear implants. Is the goal for these gene therapies to completely restore hearing and, there's no other intervention that might go with it? Or is there some way that, we might see these playing together for ongoing treatment?
Before the expert panelists and take on that question, I want to think about that concept more broadly, which is that it is normal in medicine for the first drug to come to market to be important and significant and make a meaningful change in someone's life. But it is usually never the end game, that there is a cycle of improvement that occurs, whether that means the drug could be taken less often, whether the drug has fewer side effects, or the drug has better efficacy over time. And so I think an expectation that the first gene therapy for any disease will be the last gene therapy is not what we're shooting for. We are shooting for the first gene therapy to make lives better, and then the second, third, fourth generations to make them even better, whether, as I said, it can be around safety, it can be around efficacy, it can be around costs. So in general, we expect iteration in the development of gene therapies. Specifically for hearing loss, I will defer to the other panelists about how these might be merged with other interventions at this point.
From my perspective, it's a complicated question, right? I think it depends on the type of hearing loss. One thing with congenital hearing loss and profound loss at birth, there's always, for Ashwin-C, there was always the consideration, would we be able to make a difference if we treated after child was born, and then the barriers of treating in utero and risks and things like this. So there was always this sense that it would be an adjuvant to probably also needing others. But one thing we've learned in working in the animal models is that we've been continually surprised at the resilience of the inner ear. And I think that gene therapy trials are showing that as well, that there could even be recovery and such. Certainly the timing of intervention, I think, is going to make a big difference in the efficacy and whether or not this is going to help or really be the full game. But I let other people weigh in as well.
I just wanted to add on top of both of those comments that should full restoration of physiologic hearing be the goal? Absolutely. Absolutely. That should be everyone's goal. I think that's what we're all working toward. But I just so appreciate Dr. Porteus's comments that's hardly ever where you start out. And that doesn't mean that first therapy or first set of therapies isn't successful. It is a road to success that we're on. I will say, I also, Dr. Hastings, I pulled up from your answer this idea that in part it will depend on what causes the hearing loss and what the extent of hearing loss is. So perhaps one could imagine some of the early gene therapies, if you are born completely deaf, being able to get you to a state where a hearing aid then could help, could be helpful. Whereas before, just amplifying the sounds, which is what a hearing aid does, wouldn't have been. So you certainly could see some of these existing interventions be added on top of the gene therapy. And there's precedent for that in other therapeutic areas.
Thanks for that. I think Another question from the chat, and I'm not sure, this one we might have to follow up with this person after the fact, but talking about being a carrier for the GJB2 gene, is that one that you all are familiar with and what the kind of status is for research there?
Sure. GJB2, I'll happily chime in first to say the GJB2 is the most common genetic variant that is a cause of congenital hearing loss. There's a lot of work that's going into GJB2. I really appreciate whoever asked this question. That work is not yet in the clinic. It's not in clinical trials yet, but do not take that to mean that there is not a lot of work and a lot of focus going into the GJB2 gene. There is. Lily is in that space, but we're not lonely in that space by any means. So if you have more specific questions, please reach out through ASGCT. There's a wealth of work ongoing.
I would also say, again, just more broadly, is the issue of newborn screening for genetic diseases. People are really thinking a lot about that, and the NIH and NCATS is leading a pilot project around actually doing gene sequencing at birth as a unbiased way to identify people who might be predisposed or have genetic diseases. It's a pilot program. We don't know how well or not it will work, but clearly, this idea that for most, if not all, genetic diseases, the earlier you catch them and the earlier you intervene, the more likely you are to have success. And so, newborn screening for genetic diseases is really part and parcel of developing better therapeutics as well.
This is Janet. I've been mulling over the question of the previous question about interventions supplanting other things that families are doing. In the here and now, families are questioning, for example, with the clinical trials for otoferlin, you can't introduce that to an ear that has a cochlear implant. So families, when they're finding out about this, are asking the question, should we delay other interventions to wait for future outcomes? And if we think otoferlin is a very small slice of the population, the JGB2 is much larger. So the questions surrounding this in what families need to do in the here and now with this critical window of language opportunity is families need to make decisions about what's available now for their children, and really should be in terms of thinking for our organization, in terms of thinking about that informed decision. And I'll just close with a little bit of what I started with in terms of thinking about what our final goal here is, and remembering that deafness is considered by many not a medical disease or a condition to be fixed, and that there is a thriving, beautiful community that embraces the idea of deafness as a difference within our world that makes it a better place. And I can tell you my own daughter's experience isn't black and white. It moves between those two ideas of any intervention that can help her better in her daily life and having a beautiful sense of who she is as a deaf individual too. So when we talk about the complexity of just the medical space, it's this whole conversation obviously is very complicated for a lot of different reasons. But anyway, I think about who my daughter is today and the final goal for thriving, successful, beautiful individuals in this world. So thanks.
That's so important to remember. We're down to our last minute, so I will, if you can, maybe like a 30-second, looking ahead 5-10 years, crystal ball, what would meaningful success look like for folks in the hearing loss community, for research, for policymakers to be thinking about in this field? Maybe start with you, Janet, wrap around.
Yeah, for continued funding to answer the questions that families are particularly asking about this is it safe? When will it be available? How much will it cost? What we already talked about, should I delay other interventions? Are there other emerging therapies for other gene conditions, which we've talked about? So the critical need for continued funding and research so that these questions can really be effectively answered.
Michelle?
Yeah, I have to echo that. I'm really excited about the technologies, the editing, gene therapy, ASOs. I really believe that even with what we have now, we can make a big impact on on what we know about hearing loss, but also getting a better hold on the whole entire genetic landscape of what is causing hearing loss through genetic testing. And it's just repeating that's so important and exciting to think about.
Kristen?
I think where policymakers are uniquely positioned to really be supporters is in creating and sustaining a really thriving ecosystem for this kind of research, for research to turn into drug development, to turn into cures. So think about basic scientific funding, think about predictable regulatory environment, think about access that really is tied to the value that patients and caregivers see.
And Matt?
Yeah, when I've seen videos of some of the people who've received gene therapy for genetic hearing loss and now see that they can hear and talk and interact with their families, it's invokes tears in many people. So my success in 5 year is simply, for me, is one word: more. We need more patients, more diseases. We need more approvals. We need more trials in the pipeline for this condition. So more.
Thank you. So yeah, we are at time. Thank you to all the panelists today for sharing your expertise. Thank you to our attendees for your attention over this lunch hour. If you'd like to hear more about anything you've heard today, please don't hesitate to reach out to us. We can be reached at advocacy@asgct.org. And we would be happy to share more information with your office about gene therapy for hearing loss or anything else in that same cell and gene therapy space. So again, thank you so much, and we hope you all have a great day.
Thank you.
Thank you.